Okay, great. I guess I'm on. So, yes, this is the first of three on cancer bioinformatics. So it'll be different from the tutorials you've heard already for a number of reasons. One, we have a workshop coming up in 2 weeks. So there's the first workshop is on computational cancer biology. So our goal in this overview is to kind of get people oriented for some of the research they'll hear in the workshop. So we'll try to give an overview. Second, it being cancer, we're going to have to give a little bit of the biology background. So there'll be perhaps a little more biology and maybe a little less algorithms than some of the other tutorials. And then third, as you'll see, that there's two names on this slide, myself and Nico Bierenwinkel. And at least for the first two lectures, we'll be doing them jointly. And we heard about the flipped classroom yesterday. So maybe in that tradition, if at any point you're bored with the current speaker, raise your hand. And then if the majority of the audience has their hand raised, we'll swap. We won't know what the next slide is. So I've been tasked with giving the first part here, and then you'll see us swap a few times. Let's start sort of at the beginning to get people oriented. I would say, we first want to maybe define cancer as best we can, and the first molecular characterization of cancer is due to Boveri, or widely credited to Boveri, which is that it's a disease of scrambled chromosomes. He discussed at the turn of the last century. Quite remarkable because, at the time we didn't know that chromosomes even, were made up of DNA and all the things we know now. But we have great technologies for viewing chromosomes over the past few decades, and we can see this. So, here's a normal cell, human cell, 23 pairs of chromosomes. Cancer cell, most solid tumors, extensively rearranged. Of course, we don't have to look just at the chromosomal level, and we can sequence DNA, as we'll discuss. So, now we would sort of perhaps describe cancer as a disease, many diseases, in fact, because they're quite different, as we'll discuss, that are driven primarily, but not exclusively, by somatic mutations. And these are mutations that accumulate during your lifetime, as opposed to mutations that you've gotten from your parents, inherited mutations, although there are cancer susceptibility mutations. But we'll be talking mostly about somatic mutations. So that's sort of more the modern view. And where do these somatic mutations come from? Well, they happen all the time. Cells divide. They have to copy their DNA. It's not a perfect process. The daughter cells get mutations that the parental cells didn't. Most of the time, those are sort of benign mutations, but occasionally they're not. And then, in addition to these sort of intrinsic mutations that accumulate during DNA replication, there are various environmental factors, exposures, UV light, radiation, chemical exposures, cigarette smoke, et cetera, that also lead to somatic mutations. So these mutations can go on a large range of scales. Of course, the chromosomal rearrangements that you saw in the previous picture, but all the way down to just single nucleotide changes. So somatic mutations, most benign, but some lead the cells that have these mutations. They have a selective advantage. They grow faster than the other cells in the tissue, and they'll start to grow, acquire perhaps additional mutations that give them further selective advantage and lead to sort of a full-blown tumor. So this idea of cancer as this accumulation of somatic mutations, and this idea of there being clonal expansions, the cell that acquires some advantageous mutation, then out competing its other cells in the tissue, leading into the tumor, is sort of widely now called the clonal theory. And this is sort of synthesized in a famous review article by Peter Knoll. Now, what's interesting about this, of course, in addition to the idea of understanding cancer as a process, is that by finding these mutations, we might thereby find potential targets for therapeutics or diagnostics. And the most famous example of that, in fact, also due to Peter Knoll, was the Philadelphia chromosome. So one of the first recurrent somatic mutations that was characterized in cancer, essentially the first. It's a chromosomal rearrangement to chromosomes 9 and 22 undergo a translocation. The breakpoints of those translocations happen within two genes, BCR and ABL, resulting in a fusion gene. So this was characterized in 1960. And 50 years later, roughly, imatinib, or its commercial name Gleevec, came on the market to target this fusion gene. So this is our first sort of targeted therapeutic. So what does this BCR-ABL fusion gene do? What does the drug do? So as a fusion gene, it actually leads to many sort of downstream changes through a signaling network. We're not going to go into all the sort of details here, but the idea being that this one mutation propagates its effect through this signaling network, ultimately leading to gene expression changes. And what imatinib does is it inhibits this activating ability of BCR-ABL. So it's been remarkably successful in treating patients with this leukemia. Many of them go into relapse. Unfortunately, a large fraction of them develop resistance to imatinib. And this resistance can happen in many ways. Additional mutations in this BCR-ABL gene can prevent imatinib from binding, but also additional mutations perhaps in that same signaling pathway that I showed could develop, or other types of mutations that might have something to do with the pharmacogenetics of the drug can occur. So this leads us to another part of cancer and that's important and that we need to discuss, and that's that it's an evolutionary process in a population of cells. So there's, it's always changing. Mutations are always accumulating. If you change the selective pressure by treating your tumor with a drug like imatinib, you might then select for mutations that lead to resistance to that drug. So sort of characterizing this process is an important part of what we need to do. So here's maybe our attempt, or I don't know if I should maybe not, well, this is one attempt, I guess my attempt. I didn't want to get you in trouble for this if this is a really bad attempt to define cancer, but it's an evolutionary process, somatic mutations, but then there's this notion that it's altering these signaling and regulatory pathways, and all of these sort of properties are important. So this is known, all of what I told you has been known for a couple of decades roughly. So what's new? And what's new is that we can start to quantify this process, and we can do this we can do this especially at the DNA level due to new DNA sequencing technologies, which we've heard about, right? So we can sequence genome much more readily, which means we can measure somatic mutations much more readily. We can use these technologies to measure RNA and gene expression and characterize other molecular changes. And the field's just exploded. So the first cancer genome was sequenced in 2008, which is right around the sort of inflection point of this curve. And then since then, there have been thousands, tens of thousands of now cancer genomes that have been sequenced. So now the challenge has started to shift to what can you do to interpret this data and to start to say things about cancer in all those contexts. Okay, so just one example of the types of data sets that we have. Many of you have probably heard of this data set. This is one of the big projects, at least in the US, the Cancer Genome Atlas, which is actually winding down, reaching a goal of characterizing approximately 11,000 samples, 11,000 samples from roughly 33 different cancer types and subtypes. This project is sort of really benefited from these declines in the cost of various technologies. So in addition to DNA sequencing and measuring mutations, measure these copy number changes, also measuring other types of molecular phenotypes, gene expression, DNA methylation, microRNA expression, and some protein phenotypes as well, RPPA, so a particular type of protein array, and then matched with clinical data. So for each sample, you have this sort of multi-dimensional view of it with these different technologies over large numbers of samples, and the data is all publicly available. Okay. So projects like TCGA and others, and sort of have in mind this sort of future, this personalized medicine or precision medicine or whatever the latest buzzword is for it is, where the cancer patient comes in, you would sequence their genome, you might characterize their transcriptome with RNA-seq, you might do a bunch of these molecular profiles, you might get a list of say the cancer-associated mutations, you would then have some idea of which drugs might target those mutations, and so you would have this personalized precision treatment for a cancer patient. Okay, so this is the future. Parts of this are happening now, in larger cancer centers. For you have particular cancer types, you're enrolled in clinical trials and they'll sequence your genome. So it's actually a very fast-moving space, but there's lots of challenges along each of these steps. And many of these challenges actually are computer science challenges. And of course, I work in this area, so it seems like I'm biased, but I'm not the only one saying this. And since we're in Berkeley here, I guess I have to cite David Patterson, right, who says, it's computer scientists that may have what it takes to help cure cancer. Curing cancer is perhaps a little bit overblown, but contribute to this whole sort of thing. So there's lots of challenges. So we want to try to highlight some of the challenges in this talk. And of course, the other thing then is, there's a lot of data there, so you don't have to be sitting in a cancer center in order to work on some of these problems, not only the computational problems and the theoretical problems, but you can go actually apply them to real data in some of the largest data sets around. So it's an exciting time to be working in the area. So what are some of the challenges that come up? These are sort of listed as biological challenges, but they're really all have a computational component. First, we just bought our fancy sequencer. We've run it through tumor sample. We just want to know, what are the mutations? What are the mutations in that sample? Seems like a pretty simple question. And then we'd want to classify them. Which ones are the driver mutations, the ones that are promoting cancer growth? Which ones are just these random passenger mutations? Maybe we'd like to know something about this process. What order did the mutations arise? And then we would want to then bring this into a more understanding of cancer. What's the relationship between the mutations in the genome and what we might observe in gene expression data, or in other characterization data, or in phenotype, or in drug response, and then ultimately, which ones might be the ones that are important for diagnosis and therapy. So all the way from sort of the basic measurement through interpretation through clinical application. And at each level, there are interesting problems to be faced. Now, I should say that a couple of these questions, in fact, almost these first two, when you sequence a human genome, which an individual genome, what are the differences, which ones are important, that's actually faced by almost any disease study. What makes these problems, these first two, but all of them, really challenging in cancer is that cancer is very diverse, very heterogeneous, as we say. And it's heterogeneous on different levels. So if you measure different patients with the same cancer type, same cancer subtype, you'll see different mutations. But then, remember, it's this process happening in a population of cells. So even within your tumor sample, you'll have cells with different mutations and potentially different phenotypes. So you have these heterogeneity, this difference on all these different scales that really start to challenge all the problems on the previous slide. So,
is it different from other multifactorial diseases? I mean, this heterogeneity.
So, this idea that different individuals have different genetic components is probably true for a number of complex diseases, for sure. This idea of the intracellular heterogeneity, perhaps not as much. There's maybe some evidence in some diseases, but certainly not at this idea of somatic mutations in each cell as being different. But yes, the sort of heterogeneity across the disease is certainly there. Okay, so this was the outline for part one. And there's two more to come. But for the first part, we wanted to sort of give you an overview of the different types of mutations, how you detect them from data, a little bit about sort of the interpretation, the sort of idea of which ones are driver mutations, which ones are passenger mutations, and a few different ways people try to do this. And then this third one of other topics. This is very mutation focused. The other topics are not mutation based, and we'll sort of leave that as a mystery for the end as to what those topics will be. Look at this. I'm done. And now here's our switch, so we remember.
Okay, I will continue right away with measuring mutations, and that means for us detecting mutations from sequencing data, from DNA sequencing data. So the name of the game is you start with the cancer genome, which is of course an altered human genome. You apply one of these next generation sequencing technologies, you get a large amount of short-read data, as we have seen several times today and yesterday already. And the first step here is, in almost all applications, mapping these reads to the reference genome. So you typically would map to the human reference genome. And the amount of data is large, and we have seen some of the techniques, some of the algorithms to do that. And I will not talk about this step further in any detail. So from now on, we assume that we have the reads from the cancer genome mapped to the human genome. So again, heterogeneity is a major problem. So the starting material really is not a homogeneous set of cells, but rather is a diverse sample of tumor cells, including normal cells. And all the reads are obtained from these different cells. And after mapping them to the genome, there's different things you can infer from these data. And there's two main signatures in the data. One is obviously the composition of the read. So if there's a change in a base pair, for example, relative to the reference genome, that's an indication of a mutation at this point. And the second signature is what they call depth of coverage. So you look at the amount of reads that you find in a certain region of the genome, something that Leo just called counting this morning. So if the counts are decreased, for example, like in this region, that may indicate a deletion or a similar event. Usually this is done for the tumor sample, as I have indicated here, and at the same time, in order to compare for a normal sample from the same individual. So typically when you take the biopsy of the tumor, you take also biopsy of normal healthy tissue, and then you do a comparative approach. You run the whole pipeline for both of these samples. So what are the mutations, or more generally the genetic aberrations that we expect to see in cancer? Now, it turns out that there's a large number of different genetic aberrations that have already been described. There's a long list, and this is really only a subset. And it ranges, as we have seen already, from very simple changes like a single nucleotide variant to very complex aberrations that have these fancy names like chromothripsis, Cartigas, and so on. So let me look at a few of them. So I think a single nucleotide variant is clear. This is really just a change of a single base pair, and that can be only on one copy of the chromosomes, heterozygous, or on both copies if we start with a normal diploid genome, so that would be homozygous. Then a very common event is a loss of heterozygosity. That can happen in different ways, but the most common way is just to lose a part of one chromosome, as indicated here. There are, of course, deletions essentially at all sizes. They can occur from very small ones to very large ones. Insertions can, of course, also occur. Inversions, so you can have a change in the direction here. And then, we have more complicated stuff already. Duplications, amplifications, translocations are very common. Ben has mentioned one already. And these very complex aberrations down here, they usually affect large parts of the genome, or at least of a single chromosome, and usually mess it up in a more or less complicated way, like chromothripsis, for example. Essentially, a large part of the chromosome is shattered into pieces and put together again in a seemingly random way. So this is a single genetic event that creates a big mess in the genome. So the bottom line here is that we have a lot of changes that occur in cancer genomes, and we would like, ideally, to detect all of them from the sequencing data. So far in this course, we mainly looked at SNVs, and we will also look at them here, but we will also make an effort to look at some of these other changes because they do play an important role in cancer. So let me start then with the single nucleotide variants. Again, this is a mixed sample we start with. The gray balls here might indicate normal cells, and the colored ones are different tumor cells, and different meaning that they are genetically different in one or another way. And I'm looking at an ultra-deep sequencing scenario. So we're sequencing with very deep coverage in order to be able to detect mutations at very low frequency in the tumor. Remember that we have a mixture here, and we would ideally, again, like to detect all the SNVs at any frequency in the tumor. So again, after mapping or aligning, the reads look like this. And indicated are here the SNVs that occurred on these, the blue cells have these two SNVs and so on. And on top of that, you have sequencing errors. No matter what technology you use, there will always be sequencing errors, and they are indicated in black here. So calling now an SNV simply means looking at each column of this alignment of all the reads and deciding whether the variant you see here, whether that's more likely to be a true biological variant or more likely to be a sequencing error. So this, for example, is a true one here, but it's kind of rare, so it's maybe not obvious. And also, if you look at this column, for example, these are all sequencing errors. But if you look at them, you don't know in advance. So that's really the name of the game, to distinguish and to have a model to tell you how to do this. Yes.
one sample, or you analyze a pool of several samples?
I will analyze here, I mean, right now I'm showing you like one sample, but the way I will apply it is comparing to the normal tissue. But one tumor sample, one normal. There are extensions, of course, doing it for multiple samples at the same time. I will not go into it. Oh, sorry, yeah?
Do you sequence like is a single cell type sequencing?
No, I'm not doing single cell sequencing here. We are taking the bulk and we're sequencing it with very deep coverage. Okay? But nevertheless, it's a very good question. The point is, of course, that each read here comes from a single molecule in the original preparation. So this is an important difference to Sanger sequencing, where you would just get the consensus of everything per site, and that would not allow you to do anything like this, right? So the point is that you really have a sample from the population. Okay, so some challenges when trying to do this. So the first one that I cannot make more precise now, but really most SNVs are expected to occur at low frequencies, and that has to do with the evolutionary dynamics of a tumor cell population that we will look at in more detail later. But from all what we know and from all what the models tell us, most likely most of the variants will be present at very low frequencies. So that makes it very challenging to detect them. Second one is there are these errors, and as I indicated on the previous slide already, they tend to be non-uniform. And that makes it very difficult to distinguish them from the true biological variants. And the last point is that, at least the way I pose this problem now, is that we look at each column separately. So if you do this for many positions, maybe even the whole exome or even the whole genome, you have to test many positions. So you need a statistical test that's very conservative, such that you have a handle on controlling false positives. Okay, so one common approach to do this uses in one or another way a distribution that gives you a good model of the null distribution of the variation that you observe in the data. And as one example, I'm showing you here the beta binomial distribution. So first of all, if you look at a single column, and let's say we only look at one of the bases, and we wonder whether that is a mutation or not, then the data is just binary, right? You just have a 0 if the mutation is not present in the read, and you have a 1 if the mutation is present on the read. So you can just count. All you can do basically is count how often have I seen this. So the most natural model for that, and simplest model, is a binomial model. So here's a binomial with a mean and a variance. It turns out if you just use the binomial model with fixed parameters here, you will underestimate the variation that's present in the data. So therefore, people use beta binomial or negative binomial or some sort of distribution that allows you to model more variation than you would expect in the binomial distribution. So for beta binomial, that means that this term here, this parameter here, is actually not fixed, but it's drawn from another distribution, in this case from a beta distribution, which again has a mean value q here and a hyperparameter alpha that controls the amount of additional variation. So this beta binomial distribution is very similar to the binomial, has same expected value, of course, and the variance has this additional component here, where you see where this value alpha gets very large, the additional component in the variance will disappear, and if alpha is very small, you have an additional contribution here. So let me first show you why you need such a model. So this phenomenon is called overdispersion, and it occurs basically in all of these next-generation sequencing applications. So on the left-hand side are two replicate measurements of a virus called phi-X, and plotted are the nucleotide frequencies that are estimated after the SNV calls. This is just the first replicate and the second replicate. These are all the data points. And the alpha that we would estimate for these data is very large. It's on the order of 1,000 or so. On the right-hand side is the situation we are really interested in. Here's the allele frequency in the normal sample, and here's the allele frequency in the tumor, right? And as you see, there's more spread at these low frequencies. This is double logarithmic scale, right? There's more spread at these low frequencies, and in particular, there's many points up here that would, under a binomial model, give you a call. So all of these, many of the low frequency, but including many of higher frequency variants, would end up being called, and they would end up as false positives. And here we have a smaller value of alpha. So the reason for this is that in the whole experimental pipeline, there's much more going on than just sampling. I mean, binomial distribution just accounts for sampling, but there's much more going on here. And it's extremely important to account for this, otherwise you end up with huge amounts of false positives. Okay, so here's how it works then in the end. As I said, we take the tumor sample and the normal sample, and the assumption here is that there are no mutations in the normal sample. That's strictly speaking not true, but that's the assumption we make here. We apply the same model for both of these samples, and they differ only in this parameter, which is the mean rate of generating variation. And in the tumor sample, that rate is due to sequencing errors and true SNVs. And in the normal, that mean rate is only due to sequencing errors. So then the test is simply to check whether these two are the same or whether this value P is really larger than Q. And if that's the case, then we call the single nucleotide variant. So for example, you can do a likelihood ratio test to do that. Then there's a few post-processing tricks in all SNV callers, and I'm not going through any of the algorithms anyways, or the software tools anyways. I just want to mention one here. There's many more, and that has to do with strand specificity. Namely, you want to check whether your call occurs similarly on the forward and on the reverse strands. So one reason for discrepancy might be that there's a homopolymeric region here that gives rise to an error when you read this way, whereas if you read the other way, you're not encountering that homopolymer, and you're less likely to make that error. So you would call the SNV only if you call it significantly both on the forward strands and on the reverse strands independently. And then you have to combine the p-values, and there's different ways to do that. So let's look at this in a control experiment where there were only 5 clones present in a mixture that was artificially put together, so we know what the mixture consists of. If you do that and you mix at you mix these 5 clones at frequencies that are decreasing from 10% to 1%, 0.1, 10 to the minus 4, 10 to the minus 5, then you see in the ROC curve here that it works essentially perfectly up to 10 to the minus 3. 10 to the minus 4 is still very reasonable, and then with 10 to the minus 5 it starts to break down, and that is of course expected because the coverage here was on the order of 10 to the 5. So coverage means that each base here was on average sequenced 100,000 times. So ultra deep sequencing experiment, of course in a small region of the genome. I'm not going through the comparison, but I want to point out if you just take any variation that you see in the data for granted without bothering about testing, then even for this small data set, we only sequence I think 5 genes or so here, you end up with this impressive number of over 5,000 false positives, whereas these are the true variants at the different frequencies. So being on the safe side by just taking everything is clearly not an option. You will immediately be drowned in false positives. There's no way of doing that. Finally, here's the QQ plot, which is very important because only if you have a proper way of quantifying your confidence, if you have good p-values, in other words, meaningful p-values, only then you have a chance to control the false positive rate. And you see some methods, just having a p-value itself in the output of a method, of course, doesn't help. That p-value must be meaningful. Only then you have a chance to really control the false positive rate. Here's one application to renal cell carcinoma data, and I don't want to go into great detail, but this is the same type of plot I've been showing you earlier. This is the frequency in the normal control. This is the frequency in the test, and the test here is the tumor sample. And the contour lines of the test are removed from this plot, but these are SNVs that we would call. They're highly significant, in fact, and we would also detect germline variants called SNPs here in this example. In particular, if you call the SNV, you can also estimate its frequency. That's the relative number of reads that have this SNV simply at that position. And the distribution in 2 genes here, the frequency distribution of these called SNVs is shown here. And you get a sense already that most of what we're able to detect is at very low frequencies, 10 to the minus 3, 10 to the minus 4, and this is already our limit of detection. But keep in mind, the tumor can easily have 10 to the 9, 10 to the 10 cells, right? So there's probably mutations at much, much lower frequencies here, but there's no way, even with this ultra-deep sequencing approach, to get at them. So in a way, this indicates there's a lot low frequency, but we are far from seeing all of it. So in any case, if you have called the SNVs and you estimated their frequencies, you can make a first attempt from the SNV frequencies to reconstruct the individual clones in the tumor. And this is usually done in a very simple way. Here's another conceptual picture. So you start again with a polyclonal tumor. These are individual cells now indicated by the different colors and different shapes. Here are the aligned reads. Now you do your SNV calling, frequency estimation. And then the way you try to reconstruct the clones is simply by clustering the allele frequencies. So here are the frequencies, and here's the number of SNVs with that particular frequency. Of course, there's a very strong underlying assumption here, namely that if two SNVs have a similar frequency, they are very likely to come from the same clone. And that assumption is already for larger frequencies not always correct for sure. For lower frequencies, I think there's no way of getting very far with that because there's quite some uncertainty in estimating the frequencies. If we knew the frequency precisely, it would be much better. But of course, we only have an estimate. The reads are only a finite sample. So there's a lot of uncertainty here. Yes, please.
The clones come from the same donor from the same patient.
Yes.
And the clones are accumulation of mutations. So one clone would have, say, X and Y, and then the other one would have X, Y, Z. The mutations are not disappearing from the clones.
Yes, but there's also clones evolving in parallel. So you can have then here mutation X and here Y, and then this guy has only X but not Y, this has only Y, not X. Some will look like what you are saying, and some will not share, of course, mutations. And we will get back to the mutational history of this process actually in the next lecture. Okay, so finally I want to make the point that estimating the composition of these different clones... sorry, there's a question from Cenk.
And I'm not trying to indicate that you should exchange with them. Of course, if you have multiple samples from the same tumor, are you going to come to them?
We will, not in the context of SNVs, but in the context of copy number alterations, we will look at this. Then you have more power. In general, there's still no guarantee that you can resolve it, but you clearly have more power. I completely agree. Mm-hmm.
Here, the problem of bias, like amplification bias, which basically like mutation, they can have like, for example, here all those mutations they might be from the same clone, but because they have different amplification bias based on the allele, so they might show up different frequency.
Yes, you mean in the whatever sample preparation and sequencing process? Absolutely. Of course, the assumption here is that you have a sample, a statistical sample again from the tumor, right? An unbiased sample. If there's bias due to sample preparation, there's no way here to correct for that.
To build this bias model, you actually need a lot of samples that have this mutation. So that's...
Well, I mean, if you're talking about, let's say, noise in the sense that at each sample preparation,
not necessarily noise. It's just like different when mutation happens. Like let's say you have A or B allele. Even let's say in a SNP situation, it's 50-50, but they have different amplifications. So then when you sequence and when you do deep sequencing, you don't see 50-50.
yes, absolutely. All of these biases are in here. I completely agree. That adds to bias or variance or whatever you think it is. Absolutely. Mm-hmm.
So you cannot distinguish between homozygous and heterozygous mutations.
Well, let's say in a way that's a more complex question because it has to do with the copy numbers, with the number of copies that are present in the tumor. And most tumors have altered copy numbers, so they don't have 2 in the beginning. And then the question is on how many of these does it occur and how does it occur and so on. In general, we cannot, right? Or let's say you would need to have the full copy number information in order to get at this question in the first place.
It's very unlikely to have homozygous in cancer. It's very unlikely to have mutation in both haplotypes.
It depends. No, not necessarily. I mean, for a random neutral mutation, of course, I would agree, right?
So you account for physical amplification issues in this model?
I mean, here I'm talking only about SNVs.
Right. But if you have, you know, a gene that's amplified 20-fold
Yeah.
and it's mutated,
Yeah.
then it's going to look like a different clone.
Right. So let's say you do that in a region where you have only a single chromosome, or where you know the copy number state or whatever. Then let's say only then is it meaningful to pool the SNV frequencies.
My
Absolutely.
question is, do you account for that, or can you account for it?
Well, I would just say you have to account for that. I mean, I'm just saying that the general approach to get back to the clones from SNVs only, we will later look at the copy numbers, but if you have SNVs only and you want to make an attempt to reconstruct the clonal composition, then that's what people do. And it's a good point. You have to be careful in the region that you're selecting for doing that. Absolutely. More questions at this point? Good, then let me finish this part by pointing out, I mean, why is this clonal composition of interest? Well, there's probably many answers, but I think a nice one is that if you know the diversity of the tumor, the diversity alone is in a way a prognostic marker. So this shows here incidence of a certain carcinoma of the esophagus, and this is follow-up in years. And the two patient groups here are separated according to the number of clones that have been found in their tumors. So here is the patient group that has a large number of clones, so a lot of diversity in the tumor. And here's the patient group with fewer clones, so less diversity. So the higher the diversity is, the more likely it was for the tumor to progress and to become a carcinoma, an invasive tumor at the end of the day. So there's many reasons why we're interested in diversity. One is it's a prognostic marker already in itself, even without understanding the individual variants. Just knowing how many different ones there are is informative.
But that also means you caught the tumor at a later stage, probably.
For that
Right.
study, yes. Yeah, esophagus, you get almost time series data because it's often not removed. They try to sample it several times.
Okay, so next we wanted to talk a little bit about some of the methods for detecting other types of aberrations that we see, and particularly these copy number aberrations. So these already came up in the questions. So yeah, this was already alluded to in the slide earlier, that one of the signals for seeing one of these copy number changes is this change in read depth. So you know, the cartoon here showing that if you delete this segment, actually this would generally be larger, not sort of a few nucleotides here. Think of this as a larger region. Then we'll see this drop in read depth. There are fewer reads or no reads if this region is completely deleted from the tumor on all chromosomal copies. Conversely, if you have a duplication or amplification of the region of the genome, then you would see more reads that map to that position. So by looking at this read depth signal, we can try to detect where these deletions and duplications are. Of course, one thing in looking at this read depth signal is that all you're saying is relative to your reference genome, there's been a change in the number of copies, but the signal doesn't tell you where those copies are. So you're not getting any notion of the structure of the genome. So I might know that we think there's a duplication or amplification somewhere, but we don't know where that is. So of course, this cartoon makes the process seem very simple, but it gets very complicated as you try to model all the biases, many of the same biases that happen with single nucleotide variants. When you're sequencing and mapping the reads, you have these same biases. You have to then model these probabilistically. We're not going to sort of go into this, but just to say that the signal gets very noisy. So if, for example, each dot here might indicate this read depth signal binned over a certain region of the genome. And we see this sort of variation in this, and then now we need to go and sort of figure out, has there been a change in the number of copies in this part of the region? And by eye, you might sort of see that, yes, there seems to be some drop in the read depth, and so there are various algorithmic approaches that will go and try to segment this noisy signal into regions of equal copy number. So using hidden Markov models or change point models or various approaches, we then might try to convert this signal into into a signal of regions of normal copy. Here, this is shown in a log2 ratio where we're doing it relative to the normal copy 2 of the reference genome. So the ratio here, and here we see a deletion and an amplification. So again, I mean, this is modeled probabilistically, and we have to account for the various sequencing biases and use the models that were used for single nucleotide mutations, but then often binned over a larger region. So another signal that's used for copy number, which is actually somewhat similar to single nucleotide variants, is we can take advantage of the fact that we know the locations, or we can measure, because we have the matched normal, the locations of germline SNPs, SNPs, they're inherited mutations, differences from the reference genome. So in a normal region of the genome where you have 2 copies, one maternal, one paternal, if we look at one of these germline inherited variants, it'll be a difference in the reference genome. If this is a heterozygous variant, then you'll have one copy of each. And so what we'll see is that this so-called B allele frequency is a half. Roughly half the reads will contain one allele and half will contain the other. Again, though, very noisy across the genome, but this might be, this is actually data from a chromosome. Here the centromere is missing. You can see it's sort of somewhat noisy. If we then delete a part of the genome, in fact, if we delete one of these whole chromosomes, then what happens is that this B allele frequency shifts pretty remarkably, because we don't have all the germline SNPs from one copy. And so this B allele frequency, depending on if it was the maternal or paternal SNP that we're looking at, will sort of shift to 0 and 1. Okay, so we can then combine both of these signals, this drop in read depth, this drop in B allele, both being these signals measured across the genome that then have to be segmented into regions of equal copy number. Yeah?
Can you just define the B allele? Are you saying you see two different alleles, two alleles that differ from the reference?
Right. Yes. So from the reference, two alleles that are different from the reference. It's a heterozygous. The individual is heterozygous at this position.
So when you make those plots, did you take a binning approach? Like you keep this segment size constant and you move the
These plots right here are actually just individual SNPs, and they're not binned at all. Each point here is one location. And so the variation we're seeing is just the variation in coverage, right? Because it's the previous one. Well, yeah, okay. I mean, actually, now you're going to that. This is older array data. This isn't even sequencing data, but same principle. CGH arrays. It's real data.
So B allele is essentially a
Minor allele, yeah. Okay, and then just like with single nucleotide variants, we call them, but then in cancer we have this mixture problem. Same thing happens with copy number variants. We might try to determine which fraction of the cells have a particular copy number variant. We might try to actually go further and sort of ask the question of, could we take a single tumor sample, which might have sort of normal cells without any mutations, and then various subpopulations of tumor cells, and unmix them computationally. So with single nucleotide mutations, when we tried to do this, we do this by clustering, and that's because essentially at each position we have relatively small number of reads that actually tell us something about the frequency. So the signal at each particular variant of the frequency in the population of cells is the signal is relatively low, has high variance. If we think about copy number aberrations, there tend to be fewer copy number variants in a particular tumor, but there are many reads that can be perturbed by a large copy number variant. So if we delete a whole chromosome from a tumor, that perturbs many reads. Millions of reads are now perturbed. Millions of reads that were supposed to be in a certain place are not there. So what we can then do is try to take advantage of this signal. So for example, if we had a sample that was 100% tumor and no normal cells, and there was a deletion of one copy in the tumor, then we might see this sort of characteristic drop in read depth. As I would mix in more normal cells into that mixture, normal cells having 2 copies of this region, this this drop in read depth would start to decay as I start to mix in lots of normal cells. I might not even really be able to detect it. But the idea here is that this decrease in read depth for the deletion is proportional to the fraction of cells in the mixture that had the event, that have the deletion. Yes?
Isn't there an identifiability problem here where we can't tell the difference between
Yeah.
proportion of the proportion of the tumor population?
Yeah. That's on the next slide.
Oh, okay.
Hold the question for the next slide or two. There's no model here. There's just a cartoon. Yeah.
Isn't it solvable with just using single cell sequencing?
Sure. Yeah. If you have single cell sequencing with no errors and you sequence enough single cells, sure.
And lots of money.
And lots of money. Yeah. So I'd love to talk to you about that. And then, if we have more than one population of cells, just as we often have, then we can also see that these decreases in read depth in this cartoon will perhaps indicate which copy number variant is in which population. So now here's sort of an overview model, and there are many different approaches to do this, but the interesting part here for these models is by looking at copy number variants, what we can do is describe this mixture. Again, this is sort of the cartoon, but we can describe this mixture. Think of these as individual cells. Here I'm showing a haploid genome, so to keep things a little bit simpler, there's normal cells that have one copy of each of these three colored intervals. There's these tumor cells that have 4 copies of the red, so I can describe this mixture by this integer valued matrix whose columns indicate the populations, rows indicate the number of copies of each, and then I've mixed these. My mixture is mixed these two populations in certain proportions. Here, 2/3, 1/3. So then now here's where this problem comes in. So what we then measure, the read depth here, and again, the read depth will be higher in this red region, but the read depth is governed by these probabilities, which determine that if we, say, had uniform coverage over the genome, for simplicity, assume we have that, the probability that a read would align to the red interval would be proportional to the amount of red in the genome relative to the rest, right? So we have these individual probabilities, and so the whole idea here is that this read depth is governed by some distribution. Here we might use negative binomial, whatever, but governed by this parameter p, this vector valued parameter, which is determined by c times mu appropriately normalized, okay? And here's exactly where this identifiability problem comes in, because then what we'd like to do is we'd like to sort of invert this, look at our observed data, look at our read depth, and try to find the mixture. Could we separate out this integer valued matrix and this mixing proportions, say, to maximize the likelihood, for example? This becomes a very interesting problem because it has integer and real valued variables. It's actually not a linear problem. It's not a convex problem. And only this ratio of c times mu normalized is identifiable. C and mu separately are not. So in order to actually sort of figure out what the mixture might be, and the easy way of seeing that is that, here I had one copy of each of the intervals. I could multiply the whole matrix by some scalar and get the same value p, and I wouldn't see anything different in terms of my data. So we need to use some external source. We need to make some assumption. We might assume that, we think the cancer genome is diploid, copy 2 at most of its along the genome, and that would allow us to sort of get around this identifiability issue. But it's a separate assumption we have to make. So that's sort of the outline for a number of such methods. It's been done a number of different times using different data. You have to then do the probability model different ways from SNP arrays and from genome and exome sequencing data. And these models differ in terms of how they set up the model, obviously the data types, and whether or not they just try to separate out the normal from the tumor using copy number aberrations, or whether they go further and actually try to characterize these subpopulations and predict their frequencies. There's going to be more of this at the workshop and people describing more sophisticated methods for this type of analysis. And then you can combine, and I think this was a lot of the questions that were started to be asked when Nico was talking about single nucleotide variants, and if you try to infer the clonal structure just from using single nucleotide variants and clustering them by their frequency, then you run into all these issues with copy number and how many copies are there. You could try to estimate your clonal composition using copy number variants, using the methods, variations of them that I just proposed, and ideally you'd like to use use all of it, right? And then you could combine the signal from read depth. You could combine these allele frequencies from SNVs, and this is just one example of such a method that's tried to combine both. Still a lot of work being done in this area. Okay, so finally, on detection and quickly are these other events that are, in essence, don't change the number of copies, so inversions or some versions of translocations. There's no read depth signal we can use, so we need to use other signals to detect these. And most of you have probably sort of seen the idea here. If we had perhaps done paired end sequencing, so we take longer fragments of the genome, we sequence the ends of those, we map them back to the reference, then what we'll see is that most of the fragments have distances that are what we expect based on the distribution of fragment lengths, but sometimes we'll see these pairs that map to different chromosomes or whose distance is much longer or much shorter than what we expect, and that would suggest that there's been one of these rearrangements. In fact, once we have longer reads, we don't even need to use this paired read idea. We can just take a read and look at a split alignment. So here's a version of the same thing where we might have the cancer genome is here. The difference between the cancer genome and the reference genome is a deletion of this interval, and now this read that crosses that junction point will have a split alignment across the two breakpoints. And so we can, of course, if we saw one read, we wouldn't believe this, but we would look for many such reads with the same split alignment indicating the same breakpoint. And then, of course, we can, if we had copy numbers, we can now combine copy number variants. We can combine these signals. So we can combine read depth and split reads and start to make better predictions. This is, of course, a general problem, comes up in analyzing structural variants in normal genomes as well from all sorts of organisms. But in cancer, again, the rearrangements can be much more complicated. So if you have one of these chromothripsis or chromoplexy events where you're going to have multiple breakpoints clustered together in a small region, then you have to start making predictions where you might try to stitch together these breakpoints in a consistent way. And there are approaches based on sort of building graphs from the genome that have edges constructed from the links you would get by looking at the split read information. Interestingly, not much has been done in trying to incorporate structural variants as well into the problem of reconstructing clones, although there'll be at least one talk on our workshop on that topic, so look for that. All right, so that was it for detection, and we wanted to sort of move on now to the interpretation part. So now we've detected all our somatic mutations. Could we make a prediction about which ones might be drivers and which ones might be passengers? So here's the setup. We have our own mutations. We've mapped them. Here they are. So which ones are the cancer-causing ones? Of course, that's ultimately a biological distinction. So to really answer that question, we should go do an experiment. But we have a lot more data. We have a lot more mutations that we've measured than we have had the ability to go do all the experiments. So we'd like to prioritize those experiments computationally. One way to do this, of course, is if we knew detailed knowledge about the position of the genome where that mutation had happened. We knew it was in a protein sequence that was in the active site. We could make predictions about, particular locations of the genome. If we don't have that functional information, and for many positions of the genome we don't know what they do, then another idea is just to use the fact that we can measure mutations across many genomes. So we could look for positions in the genome that are mutated frequently or genes that are mutated more often than we would expect. And this would give us some indication that this might be a cancer-causing mutation if we see it more than we expect. So here's some real data. We sort of took the 3,000 patients from this TCGA across 12 different cancer types. And if you just count up the number of samples that have mutations in particular genes, you see the first gene here, P53, is the most well-known cancer gene around. So we've done pretty well. We can see that mutated in more than a third of the patients. And then we have these additional genes. The second gene, TTN, called Titin, also a very famous gene. Anyone know anything about Titin?
It's really big.
It's really big. Its muscles are huge. So this was a completely ridiculous way to look at the data by counting up samples, although you often see people doing things like this, surprisingly. But for the computational audience, you'd see that was silly. And of course, if I were to look at these genes, some of them are cancer genes. They're actually picked up a bunch of long genes. Titin's the longest gene in the genome according to certain ways of counting it. So we, of course, want to make this prediction much more precisely, and we can do that. And the simple way to model this, of course, would be to model it as a sequence of Bernoulli trials. We've sort of flipped a coin for each position in the genome to decide whether there's a mutation. So there's some rate of background mutation, and then we can count up the number of mutations that we'd see in a gene across samples and use binomial distribution and decide on some comfortable FDR and then predict that those are the cancer genes according to that FDR. So remarkably, if we just use that simple model, we actually start to do pretty well on this data. The first 4 are actually very well-known cancer genes. This one as well. A few of these other stragglers, probably not cancer genes. And that's because calibrating this background mutation rate is actually really hard to do. That background mutation rate varies by cancer type, by tissue. It varies by other sort of covariates, like the expression level of the gene might have some effect on the background mutation rate. The replication timing, when that region of the genome is replicated during DNA replication, has some effect on the mutation rate. So just getting this parameter actually becomes quite a challenge.
Also the stage of the cancer.
Yeah. So there becomes, what you almost need is you need this parameter determined for the sample that you're looking at, right? But by determining that parameter for that sample, you've almost solved the problem that you were trying to address. So there's a little bit of circularity almost that comes into this. But they're great. They're increasingly good models for doing this for that seem to work pretty well for coding regions. Non-coding regions still a little bit open in terms of how these models are predicting regions of the genome that are mutated more often than expected. But what happens in applying this model is, again, this sort of idea of intertumor heterogeneity, this process of mutations accumulating in cells is occurring independently in each individual, right? I mean, there's no, these are somatic mutations, right? So they're occurring independently each individual. They're somehow correlated by the fact that these 2 individuals have the same cancer type, but they tend not to be hitting the same genes as often as one might have sort of expected. So when you apply one of these models to predict significantly mutated genes, you often get a picture that looks something like this. Here, genes ranked according to their significance. And what you'll see is that for a sort of modest-sized experiment, you only get sort of a handful of genes that you can confidently say are mutated more than expected by chance. And then there's a bunch of genes, so-called long tail, of rarely mutated genes that we can't distinguish from chance according to the number of samples. So we can push this line longer by push this line further in this direction by sequencing more samples. So some people certainly propose to do that, but this tail gets pretty long, and you have to start pushing it pretty far to make the statistics work if you look one gene at a time. So the other approach that people have tried to do is to try to sort of understand this long tail and this behavior is, genes don't work alone. They work together in various pathways and processes or circuits. And so cancer, wanting to grow fast, for example, has many genes it could mutate, or even non-coding regions it could mutate in order to amp up its growth rate. So perhaps if we think about this complexity, we're not surprised at all to see that there is this long tail phenomenon, because what we're seeing is just the sort of illustration of all the different ways that mutations could have hit the genome to lead to that same phenotype. And that's a big combinatorial, there's a huge number of combinations in order to do that. So instead of analyzing one gene at a time, maybe we should try to analyze in these pathways or circuits and look for sort of clustering of genes. And some of the cancer sequencing approaches have done this, and they draw these nice pictures that show you somehow the clustering of mutations in particular signaling pathways here. And you'll sort of see things like RB signaling, 67% of the cases mutated in this ovarian cancer data set. There's no single gene mutated in more than 50% here. et cetera. And these are sort of pictures that illustrate this clustering phenomenon, but they don't actually show you anything new. There's no p-value associated with this picture. There's no sort of statistical significance with these pictures. So could we sort of go further if we think about cancer as this disease driven by somatic mutations, and those somatic mutations alter pathways? Then could we find the pathways by just looking at where the mutations happen in combination? Right? So could we discover them? So we've got a lot of data, more coming. So it's almost a natural perturbation experiment. Maybe we would start to be able to pick out the pathways by seeing where the mutations cluster. You can, of course, see this phenomenon. If I tell you the pathways ahead of time, you can go and evaluate whether or not they're mutated more than you expect just by taking that single gene test and applying it to the group of genes in common. But if you do that, you're not getting really the picture. You're not going to discover anything new. You won't see any new pathways. Somehow these pictures are sort of showing you something about actually the interactions themselves and the topology of the interactions, whereas these tests are just showing you individual gene sets and these interactions between pathways. The pathways in the databases can be large. They can overlap in different ways. So we'd like to do other things with the data. So could we discover the pathways by taking the data? So could we, another way of saying this is, could we reduce our prior knowledge? Instead of looking only at known pathways, could we discover them? Of course, as we look at sets of genes, there are many, many sets. So, we can't look at all combinations of genes. And when you start to think about doing this with non-coding regions of the genome, it blows up even further. So you can't essentially check all combinations, right? You just nowhere near enough data to do that, probably ever, in terms of the number of samples you would need. So is there anything in between these sort of two extremes that we could play around with? And people have done this in a number of different ways. I'll just show you two quickly. One is like somehow an intermediate between these extremes is to look at one of these interaction networks, which kind of show you globally something about the structure of interactions, but haven't sort of predefined the sets of genes you're going to look at. Or you can sort of make some hypothesis about what you expect these combinations to look like. And one of those is mutual exclusivity, which I'll show you quickly. So here's just sort of one setup of an approach that's been used. Rather than looking at a known pathway, let's take let's take a whole genome interaction network. People have constructed these things. You can construct them from well-known pathways that you superimpose. You can bring in all sorts of high throughput data, yeast-2 hybrid data, mass spec data, everything you know about interactions in your cell type of interest. And now you'll have your mutation data, and rather than test one gene at a time or test a known set of genes, could you just sort of find parts of the network that are mutated more than you expect? It's kind of what those pictures were sort of showing you. And so what you need to do is somehow pick out subnetworks according to both the score that you would assign to the vertices, which are here in the genes, for example. There's a protein-protein interaction network, and the interactions between them. So you just simultaneously consider both. And this is sort of the realm of what are called network prioritization methods. So given a network and given some labels on the vertices, can you predict other vertices that are interesting because they're nearby vertices that you've already implicated? So for example, you might have a situation like this where here are 4 genes that we know are cancer genes. Could we predict new cancer genes? So we might expect that maybe these 2 are also cancer genes because they interact a lot with cancer genes. So these approaches that look at interactions in network neighborhood, it turns out biological networks have these high degree nodes or these hubs, this very heterogeneous topology. So looking at nearest neighbors often doesn't work as well as doing this in a way that sort of encodes the topology in a more continuous way, if you will. So using a network flow approach or using a random walk or diffusion approach on the graph, you can encode the topology of the graph beyond sort of nearest neighbors. So in essence, a lot of these methods just reduce to linear algebra, really. So if you take these network propagation approaches, if you take a random walk on a graph, or if I do a diffusion of fluid on the graph, and I want to know the equilibrium distribution of that random walk, or I want to know the distribution of the probability distribution of that random walk at a particular time, or same thing for diffusion, then essentially the way I get that is by taking a matrix which encodes the transition function, so the heat kernel, the transition condition of the random walk with restart, multiplying it by the initial distribution, and that gives me the equilibrium distribution of the distribution at time t. So what I can do is interpret that as taking my scores of my vertices, my gene scores, multiplying them by this matrix that encodes the topology of the graph, thereby getting a ranking on the genes. So the simple way of sort of thinking about this is maybe this vector here is, 1 on a few nodes, 0 everywhere else. So my initial condition. In fact, maybe we should think of 1 on one node, 1 on a single node, 0 everywhere else. And then when I multiply this vector, what I'm getting is the equilibrium distribution of the random walk if I initialize at that point. So that'll give me some ranking of the vertices relative to my initial condition. And then I can use that to rank cancer genes. You could actually play this game a little bit further, where instead of looking at one initial condition, you can actually take an initial condition where you put a sort of score on each node, 0 everywhere else. So you can think of this as a source of heat on this node, and then you can look at essentially the ranking of each node relative to every other node. So instead of getting a single vector ranking all genes, you get actually a series of vectors which you can encode as a matrix, and this is really then just giving you a similarity matrix between the nodes. So the entries as a matrix, I could interpret them. For example, if I think of this model as, let me take a model where I assign heat with each of these entries h1 through hn to each vertex. What's the heat distribution at a certain time on this graph? That will be determined by the entries of this matrix. So this matrix Sij will tell me the heat on each vertex I at some time t, given an initial heat J on vertex 0, at the other vertex at time 0. Right? So it's giving me a similarity, and then you could take that similarity matrix and find clusters in the graph. And this will then give you these subnetworks that are governed by both the topology of the network, as determined by the diffusion process, and the initial distribution of scores. Yes?
So how long do you run these random walks? Because if you run them for long enough, then it won't depend on the initial condition.
Right. So this is where all the differences in the methods come in. Is what do you use for the random walk, and how long do you run it? So if you do just the regular random walk, and you run it for infinite time, then your equilibrium distribution is like the degree distribution, actually. So not very interesting. If you run it for some time less, for a few steps, then you get sort of a distribution that's looking like what's happening locally. So it becomes a parameter of the algorithm. The other way this is done is if you do something like this random walk with restart, sometimes called the teleporting random walk, where at each step you have a certain probability of choosing a neighbor. And with probability alpha, you choose a neighbor. With probability 1 minus alpha, you jump back to your starting point. This is what the PageRank algorithm is based on. Then you do get an equilibrium distribution that is, non-trivial. There, it's governed by the parameter alpha. So there's dozens of these different methods that essentially all differ in what matrix you use here, whether you're looking at an equilibrium distribution or a non-equilibrium distribution, and whether you're trying to, in the end, get a single vertex ranking, or whether you're trying to kind of turn it into this similarity matrix. Yeah? Or. Yeah, or. Another series of methods,
you can just calculate the distance as the expected length of the random walk, which can, again, be used as
Right, and that's the other way to do it that gets rid of this parameter, is that you can essentially compute these expected values, right? So there's things like the commute time, for example, which is the expected time to, to walk the talk. Yeah. Yeah. So you can then take these prioritization methods and try to use them to sort of pull out these subnetworks. This is just sort of one example that was applied to the same data set, where you pull out now not single genes that you're predicting are cancer genes, but you're pulling out groups of interacting genes. Many of these correspond to protein complexes, and you're starting to sort of see this. Another approach that uses less prior information, now no gene sets, no interaction network, is to use some signal we expect of perhaps mutations in pathways. So this one signal is mutual exclusivity. So the argument here is that if we look at a signaling pathway, what we tend to see is that there's approximately one gene in this pathway mutated per patient. There's sort of two arguments for this observed pattern. One argument is that there are relatively few driver mutations in a tumor, and those mutations have to hit different pathways, and so the back of the envelope calculation sort of gives you one mutation per pathway. The perhaps more interesting argument from the evolutionary perspective is that if you've mutated a pathway by mutating, say, one of the genes in the pathway, then a second mutation in that same pathway doesn't give the cell much of an additional selective advantage, right? So therefore, you tend to only see one mutation per pathway. Of course, there's some noise in the signal. So then what you can do is, without any prior knowledge of interactions, you can take your mutation data, here genes and tumors, and try to just look for sets of genes that exhibit this pattern of mutual exclusivity. And you can do this in a combinatorial way, where you try to find, large sets of genes that are approximately exclusive. This is sort of a binary matrix, so it's a nice combinatorial problem to find these mutually exclusive sets. Or you can go further and you can try to model this probabilistically. And the advantage here is that you can actually start to model more about the process. So for example, you can look at this pair of genes, these observed counts of these genes in terms of their mutations. Those observed counts depend on the mutation rate of each gene, but also our observation time. So for example, we might see mutual exclusivity between two genes just because we haven't somehow waited long enough. We measured we sequenced the tumor early in its progression rather than later. If we had measured it later, we would see that both mutations occurred, and we can model that probabilistically and incorporate that into the models. Wow. Okay, so the last part, other topics.
Just before your learning breakdown, I have the pleasure of talking about this exciting last point here, other topics. We basically included that because obviously we are very genomics focused in everything that we are talking about here. And we just briefly wanted to mention a few other things that either we know a lot of people are working on, or we think it is important and it might be worth working on. So the first one I want to mention is more a biological thing in the first place, and that is the formation of metastases. So this is something we haven't really talked about so far. I think it's a very important process, and there will be a lot of data coming up in this context. So what we mean is basically that the tumor progresses like this. There might be a clonal origin typically. The tumor grows, accumulates different mutations indicated by the different colors here. And at some point, cells leave the primary tumor, and some of them, usually a tiny small fraction, is able to seed a new tumor, typically in another organ of the same patient. And this process is highly relevant because clinically, that's clearly the most important aspect. In fact, 90% of all cancer-related deaths are due to metastases. So the primary tumor is in most cases not the problem really, clinically, the main problem at least not, but rather the metastases. So in a way, this is the process that, at least from a clinical point of view, we would care about most. In a way, it's a highly inefficient process, and I want to make that a little bit more precise. So here's a slightly more detailed cartoon of this process. Again, here's the primary tumor, and it typically will go into the blood system. So a process called intravasation. In the first place, these cells have to acquire the ability to leave the primary tumor. The normal tissue cells, of course, do not do that. But tumor cells, or some tumor cells, at some point in time have this ability. Then they're circulating in the blood, and they can leave the bloodstream again, a process called extravasation. And up to here, from the data that we have, this process is fairly efficient. A lot of cells seem to be able to do all of this. But then actually seeding, initiating a new tumor, this is where only a small fraction of cells are successful. So some of them will be able to replicate in a new environment, to grow and to initiate a new tumor here. So only few metastatic colonies will eventually survive. So in a way, this is a process I think we need to understand at this high level maybe, we need to understand it in terms of the stochastic processes that happens through all of these different bottlenecks here. And of course, we would also like to go back to the molecular profiles and understand whether molecular composition of these cells, from the molecular profiles, we can predict any of these steps or the abilities of the cells to go through these steps. Yes?
Is that really an inefficient process? Because many of these cells, they get to a new site and they become dormant, and they just wait for up to 20 years to reactivate.
Right, but we would like to understand why and which ones do that, which ones don't do that. Absolutely, I mean, this is, yeah. Of course, if a single cell is dormant somewhere, we typically cannot see it today, right? So there might be many of those, but...
People do, and they're starting to count them.
But those are clinically, again, not a problem, probably, as much as we know. Okay. Okay. Good. Then these circulating cells do not only occur in single cells, but also in clusters of something between 4 and 50 cells. Actually, 2% of them occur in clusters. And if they occur in a cluster, they have a much increased chance of initiating a metastasis, indicating, of course, that the environment in which they grow is very important. So if they already come in a way with their own environment, chances are better for them to see the new tumor. At least that's what the mouse experiments here indicate. So there's a number of challenges with metastasis formation. So we would like to predict metastatic potential from omics profiles. I mentioned that already. We would also like to predict the tropism of circulating tumor cells. So to which organs are these cells likely to go, or in which organs are they able to see the new tumor? In order to understand whether genomics or other molecular markers play a role at all, we would need to have a good null model here. So what do we expect? Let's say molecular profiles do not play any role. What is the chance of a primary tumor seeding a metastasis? And this is very difficult because there's so many bottlenecks, so many rare events, so the variance of this process is very high. We will probably never have enough data and so on. So I think this is a very interesting but also relevant question. And of course, we would also like to understand the evolutionary history. If you have multiple metastases, do they all come from the primary tumor, or do they depend in other ways on each other? Has one metastasis given rise to another metastasis and so on? Many tumors have, unfortunately, the property of spreading a lot in different organs, and we would like to understand how they depend on each other. And we will talk more about this general topic after the break. So a few other things I want to mention just on one slide very briefly each. Obviously, as I said, not only genomics data are generated, but a whole range of other omics and in fact also other data types, for example, epigenomics data, transcriptomics, proteomics, metabolomics, and so on, often called multi-omics. And obviously, you would like to integrate all of these data and not analyze them separately. This, I think, is a huge challenge where also algorithms are very much needed in order to integrate these data and eventually to make predictions based on many of these profiles. One such subproblem has, in fact, a longer history, and that is tumor subtyping, because that has been studied quite extensively already in the microarray era when the data came up. Transcriptomes have been measured with microarrays. When people started clustering and co-clustering these data sets where the expression of many genes, genome-wide, let's say, have been measured in a range of different samples. Ron and others have done a lot of nice work on this. So I think extensions of these types of approach, incorporating other molecular profiles, are very much needed. Finally, I want to come back to treatment. So in the first place, there's also an increasing amount of drug response data that is generated in more or less high throughput manners. So the data is of different types, but one is that you're measuring the response to a number of different drugs when perturbing a range of different genes. There are assays doing that in a genome-wide fashion. So from these data, you would like to learn a model that can predict the response to a drug from the molecular profile, for example, from the mutational pattern that you found in the cancer genome. And eventually, these predictions should be to the benefit of the patient. So personalized medicine, or sometimes called precision oncology in the context of cancer, is in a way one of the underlying goals here, and from an application point of view, maybe the main underlying goal. And really that means taking these molecular profiles and using them for clinical decision making. So for cancer, clinical decision making is typically made in the tumor board. This is like a circle of experts sitting together discussing the cancer cases, at least the more difficult ones, and then deciding together on the treatment. And the tumor boards these days, they have a genomics and informatics component. So we really need computer science and statistics in order to inform the tumor board and to make the best possible predictions for the patients. So some of you or some of your students will eventually sit in a tumor board and will have to give advice to the treating physicians how to set up the treatment and help finding the best treatment. And it is important for that process to understand the data and to understand the limitations and the possibilities of analyzing the data. With that, I would like to stop here and just reiterate that computer scientists, so that means you, may have what it takes to help cure cancer. And we both take questions. Yes.
So, in terms of this detection, you talk about right? Is there a specific reason?
Insertions?
Yes.
You mean like novel sequence insertions? Because, so in cancer, I mean, often, in looking at human genomes, you might look at novel insertions that weren't already present. Mostly when we're looking at somatic mutations, there isn't new material being inserted with the exception of viruses. So there is work, we didn't put it in here, there is work in detection of viral insertions in somatic tissue, and those are important for certain cancer types. Yes, and also retrotransposons are hopping around in cancers all the time. Yeah, pretty much any mutation probably that can occur will occur in cancer cells because they generally get very messed up genomically. So, I mean, in terms of detection, I mean, you could take any of the methods that you use to detect a class of variants that you apply to human genomes and apply them to cancer genomes. And, that will work pretty well depending on sort of the purity of the tumor, right? So as you start to have these admixture problems with normal cells, or you start to have multiple subpopulations, that's where things get different in the cancer context, and you sometimes have to adapt the methods, or you can, do more interesting things with the methods. You can start to predict subclonality.
Yeah, just want to note that if you look at many of the cancer genome annotations, I can tell you the vast majority of the novel insertions claimed are actually interspersed duplications or other events. You have to check the study.
Yeah.
Well, I actually think that the big problem with detecting insertion is when this insertion, in terms of this large range of insertion, the short reads actually have limitations to figure out those large range of insertions.
Right.
So large range of translocation. Anything is a large range of structural variation.
Yeah.
Yeah, short reads have inherent limitations to discover those. So actually, I'm thinking. Not thinking. So why don't we try long reads that can cover a whole range? We didn't have the technology, but we have.
Yeah. No, and there's certainly some work that people are doing with PacBio and 10x and these virtual long to get better things. The other point we didn't say though, and I think your insertions reminded me of this, is that small indels are actually still quite challenging with short reads. So things that are, in the range of 10 to 80 nucleotides are also tend to be quite difficult to detect. And there was just a paper a few weeks ago about very complicated indels that happen in tumors where there's sort of lots of shuffling of the bases and more complicated indels than people have seen in germline, and so you again need specialized algorithms to try to detect those. So that's also an area that's been challenging.
But actually, I think this whole community actually rely on this mapping.
Yeah, sure.
Mapping. The thing is, not really many mappers are good at heterogeneous cases.
Yeah.
Actually, fundamental issues.
Yeah, no, and I think that, of course, one could attempt to assemble cancer genomes de novo without mapping. That you run into all the usual challenges you have with de novo assembly, then compounded by the fact that you're not assembling one genome, but potentially many. It's certainly of interest, and as the data gets better, people are doing that. I'm not aware of anyone sort of doing de novo assemblies. It's essentially meta genome assembly, but where, yeah.
We heard yesterday that's problems completely solved.
Exactly, yeah. And then single cell stuff we'll have in the next hour. So there's single cell came up, but we'll have some. Yeah. Any other questions on this part? I know we kind of
there's one over there.
Stuff.
During metastasis, how is genetic information transferred between cells?
How is so? I mean, if the end. The question was how is actually I didn't quite understand, but the question is how is genetic information transferred during metastasis formation? No. Yeah. Okay. I'm not sure I understand the question, but the whole.
How metastasis works.
Yeah, but there is a tumor. I think it's conceptually very simple. So a tumor consists of many cells. And each cell has the genetic information, right? And so the entire cell leaves the tumor with the genetic information, and then here is the genetic information in the nucleus.
So then, at that final step, does there have to be any information transferred between the cancer cell and the native cells in the new tissue?
I mean, these are only cancer cells in my cartoon here, at least, and this just indicates that this cell is replicating, it's proliferating, and therefore there's many offspring then at some point. It's the tumor again growing, starting from a single cell. This tumor at some point started from a single cell, and one cell, many cells left, but one was able to again initiate a new tumor at a different place, a different organ.
But if I could add one thing, I mean, I think one thing you pointed to was, are there other, does it matter what the other cells are? And there are lots of interactions between non-tumor cells and tumor cells, and the whole sort of tumor microenvironment and what the interactions are between these cells. I mean, there's all sorts of signaling molecules that are, going across these cells is a huge and important issue. One we didn't touch on. We won't really be touching on it in the workshop either, unfortunately, but it's still a big area. Is the tumor microenvironment, how that might contribute to metastasis, I think is still a very open question.
Yes.
So, as I understand, the main assumption is that cancer is due to either mutation, insertion, or deletion in the. So.
You mean the assumption of the entire tumor progression process?
Maybe just the way it appears or the way it develops is due to a set of mutations?
Yeah, I mean, we took, as I said, a very genomics-based point of view, and I probably think that most of the changes are due to changes in the DNA, but there are certainly also epigenetic changes. There can even be changes to the transcriptome without, or to the proteome without having an underlying change in the DNA. And all of that, I think, will also occur. I don't know. I mean, in terms of the epigenome, there's quite some data by now. The other things are maybe a little more subtle. So it's true in a way that the DNA is what we can, first of all, measure easiest. and probably also what's most informative about the evolutionary process. I think that also most phenotypic changes of a tumor cell are due to genetic changes, but it's not clear, and definitely other things will play a role here.
So why is metastasis responsible for 90% of the deaths? Is the tumor formed in metastasis more aggressive than the original one?
I don't know. It's probably the best. I mean, it depends. It's probably a good answer. Do you have an answer?
It colonizes organs and causes organ failure, for example, the liver. That's a perfect example.
But sometimes the original tumor is already in an organ where it could cause that.
Yeah, but
individual
those
tumors
can generally be resected. It's the metastatic.
they tend to be
self-limiting
constrained by
too,
blood
right?
supply and so on, and so new locations and got more available blood supply to go off and so on.
Here's one additional theory. Maybe the vast majority of the non-metastatic tumors can be treated by surgery.
Yes?
You mentioned Gleevec as inhibiting the source of the cancer, but then you mentioned that usually mutations are accumulating to a pathway. Is there an attempt to find like the source of accumulation or the downstream of accumulation and then treat it instead of the actual?
I guess the point was that the drug targets this particular fusion protein, but the tumor, essentially against all of the drugs that we have, the tumors develop more or less rapidly resistance, and that means that if you think of the signaling pathway, they find other ways of avoiding apoptosis, increasing proliferation, and so on. And then the name of the game is, if there's another alteration, can you treat that with another drug, or can you avoid it in the first place, and so on. And that is not overly successful so far. People start playing with combinations of drugs rather than giving one. I mean, it's already done in some cases, but let's say there are some hopes that if you use drug combinations, you have more selective pressure on the tumor in order to avoid that. But in general, it's drug resistance development is a big problem, especially for the targeted treatment. So not the general chemotherapy, but all the targeted treatment. Drug resistance typically develops very quickly, within months or a few years.
Should we go to the break?